Assalamualaikum and Selamat Sejahtera. We are from Group 7 and now we're going to present about data logger in aerobic respiration experiment.
I'm Lo Chen Hui. First of all, I would like to discuss some of the importances of the implementation of data logger in science education. Data logging is the process of automatic capture of data in an experiment. By using data logger in science teaching and learning, it helps us to collect data automatically from the installed sensor for a preset duration and interval. Without the need for personnel to spend time taking and recording measurements, it can help us to improve productivity by saving time. The use of data logger in school also increases students' interest and motivation in science classes, as students are able to link the concept learned with the data collected from the data logger during laboratory work. Next, our group would like to introduce the experiment on aerobic respiration of peas that can be performed by students and teachers in school. Data loggers are used for the data collection in the experiment. In this experiment, the use of oxygen in the aerobic respiration of peas can be seen in a sealed container, which the level of oxygen reduces with time. Before starting the experiment, teachers can point out some questions to engage students about the topic. Example, do plants breathe? Plants use oxygen during aerobic respiration. However, unlike animals, they use organelles called stomata and lentic cells for gaseous exchange. The next question is, when do the plants undergo aerobic respiration? Aerobic respiration occurs on both day and night. During the day, plant cells can use some of the oxygen produced by photosynthesis for respiration. However, during the night, photosynthesis stops and respiration carries on.
My name is Farha and the next one is Empower Phase. In this experiment, we use the RDAQ as our data logger. The RDAQ is a versatile instrument that connects to the USB port of any PC. It has 3 sockets that are dedicated to external sensor. In this experiment, we use oxygen sensor as our external sensor that will be connected to the socket labeled Exit 1 on the DRD. The last column shows that how the experiment set up. The next one is the result of this experiment. As the time increases, the number of oxygen will decrease. In this phase, there will be a few questions that will be asked to the student to increase their understanding about this experiment. This is the example of the questions. The first one, by using the ideal gas law and the experience from this map, give the variables that had to be controlled from your data. The answer is ideal gas law by using the formula PV equal to nRT. The second one is, what result would you expect if you design an experiment to examine the rates of cellular respiration with peas that have been germinating different lengths of time, 0, 24, 48, and 72 hours. The peas that were germinating for 0 hours will have the lowest rate of cellular respiration. As time goes on, rate of cellular respiration increases. The peas that were germinating for 72 hours will have the greatest rate of cellular respiration.
Okay, we move on to the next part, which is the enhance. So before, I would like to tell you about the importance of this aerobic respiration experiment. I would like to share some the specialty about this instrument. So this is what we found on the internet. The managing director of Pico Technology once said that the new updated Dr. DAQ means that we now offer a complete range of USB measurement products from small, low-cost data loggers through a high specification oscilloscopes. And as they all share the same Picoscope and Picolog software, it's easy for our customer to upgrade their hardware without having to learn a new user interface. He said that. So it is important in our daily to record various experiments specifically for this experiment, which is the aerobic respiration. So we might also can investigate to see if light intensity has any effect illuminating the piece with a light bulb, and additionally record the light level. And apart from aerobic respiration, this Dr. DAQ also can be used with the pH probe to find the pH of various indigestion remedies and the effect of adding HCl, dilute HCl, which is the dilute hydrochloric acid. So the principle is that the stomach produces HCl, and this acid provides a medium for digestion of protein of the enzyme by the enzyme protease. So that's the other specialty about this instrument. So back to this back to the importance of aerobic respiration experiments. Of course, this instrument is important for aerobic respiration experiment because it produces more ATP. And here's some brief explanation about the aerobic respiration. So the cells metabolize sugar and nutrients to provide themselves with energy. In the mitochondria, cells undergo cellular respiration, where the glucose that we consume from foods is broken down by many cycles through a glycolysis process, Krebs cycle process, and many other processes that is related about this aerobic respiration. Okay, so this is the actual process of aerobic respiration, and I believe that we all have learned this before. This is not something new we have seen into. So I will not go through it, or I will not explain detail about this, because we have learned this during our matriculation level, or some of us is from foundation science and form 6, and I believe that we all have learned during we are in that level, during in matriculation life, or foundation or form 6, right? So I will not explain detail about this, but I will briefly explain again about the aerobic respiration. So aerobic respiration is the aerobic catabolism of nutrients to carbon dioxide, water, and energy, and involves an electron transport system, which is the ETC, in which molecular oxygen is the final electron receptor. So most of the eukaryotes and prokaryotes use aerobic respiration to obtain the energy from glucose. So we have to remember this chemical formula or chemical reaction, which is the glucose C6H12O6 plus 6O2 will react with each other and will produce carbon dioxide, which is the CCO2 plus CH2O.
Hello, my name is Easter Julian, and I'm going to continue the presentation. As my friend has presented earlier, it is very convenient to use the data logger, which is DRDAQ data logger, which is used in this respiration experiment. It is very convenient because it gives accurate data and result, and it enables the data and result to be transferred directly to the computer to be analyzed directly. So it is easier for human to conduct experiment, especially school students. So here we can see there are two figures showing a traditional method of doing the respiration experiment, and this one is the new method of conducting experiment of respiration. So in the traditional method, students or the one who conducted the experiment has to observe the number of air bubble that is present throughout the experiment and count the number of air bubbles that is produced throughout the experiments to indicate the level of carbon dioxide or oxygen that is being investigated in the experiment. Whereas in this new method, a data logger is connected to the experiment itself, where the data or the level of oxygen or carbon dioxide is directly recorded inside the data logger itself and transferred directly to the computer so that it is easier for us to analyze the data and makes a graph or a graph or a chart out of it. Okay, so this comparison shows that the data logger, the usage of data logger really makes things easy for us to conduct experiment, especially in this respiration experiment. So in conclusion, this data logger is very conventional and convenient because it will automatically monitor and record our environmental changes in the experiment and save the data to a computer connected to it, making it easier for us to analyze the data. This will help in reducing the human error that we usually do. So it is our job to help in showing that it is a good idea to acknowledge the convenient or the usage of data logger in school, especially in the implementation of STEM education in Malaysian school. So it is a very good step or effort in promoting the usage of technology in terms of data logger, where the students or the teachers can be exposed to data logger, where they can conduct experiment easily and effectively. So that's all from me, and that's the end of our group's presentation. Thank you.